And thank you very much for joining this seminar series. Just reminding you that we are coming from the IU School of Medicine Center for Medical Genomics, which is the genomics core facility for Central Indiana. And we work very closely with other genomics core facilities at IU Bloomington, Purdue, and Notre Dame, and also to a large extent with Ohio State University as well. The service of our center include mostly on the next generation sequencing, single cell analysis, as well as spatial omics. And there are quite a lot of recent acquisition for different high-end equipments in our labs. For example, we just got the NextSeq 2000 installed in the lab, and we will have a CosMx system that coming in for the high resolution of spatial omics. And then we are going to have Illumina NovaSeq X Plus, which is the brightest, the newest, the highest throughput sequencers that they are currently on the market, and that will be installed in our facility very, very soon. And with all those applications, we can sequence whatever we want to sequence, but by the end of the day, come back to the biology we want to answer. And the library preparation is such important elements, and that's the reason that we're inviting Twist Biosciences to come here to give a seminar. And Twist has been doing a lot of things, and I think Tim and you will tell us more about that. But they recently have this new product on the methylation side, which we did some of the piloting in our own laboratory. It's very, very interesting data. And we're very excited about that. So I think it's a great idea to have Twist to come here to give a seminar introducing this capacity to our investigators. Hopefully that we will be able to take advantage of it. Our speaker today is Dr. Tim Fahrenheit from Twist. And we have been interacting quite a bit on the Zoom and with different settings, and she will give a very, very interesting introduction. Without further ado, thank you very much, Tim. Look forward to the talk.
Thank you very much. Thank you everyone for attending and for the invitation to come talk to you about a relatively new product that we released and we've been working with the CMG very closely with and it's our methylation detection system. But to start, just so everyone's on the same page, I just kind of want to introduce Twist, since we are a relatively new company in the NGS space, and a little bit of background about target enrichment in general, and kind of the metrics and things to look for that are indicative of a successful target enrichment experiment. So Twist produces DNA. So that is our core technology. We do it in a little bit different fashion than most other companies. So instead of producing it in a 96-well plate where each well would be a given oligo, we have a silicon-based platform that can print DNA at a much higher throughput at scale and a higher quality with lower error rates. So with that core technology, we apply it to anything that could use DNA. So we have a synthetic biology side where we do clonal genes, where we'll clone large gene fragments into your vector of choice, along with single-stranded oligo pools, combinatorial variant libraries. We have a data storage department that is storing data in DNA, and then a biopharma division that is doing high-throughput antibody screenings, humanization, and those sort of products. Today, we're going to focus on the NGS side of the business, since I'm an NGS FAS, and we're talking about the methylation detection system. And we leverage this high throughput oligosynthesis technology to create target enrichment probes. So target enrichment has been around for a while. It's how we generally do whole exome sequencing. And the whole idea is that if you're only interested in a subset of your genome of interest, you don't need to sequence the entire genome. You can reach in with these probes and pull out just regions that you're interested in, dramatically lowering the amount of sequencing depth or sequencing reads that you need to dedicate to a given sample. There's an example of an exome where at 20x, it requires 7.5 gigabases compared to a 20x whole genome sequencing where you would need 90 gigabases. So a significant reduction in the amount of space. And this allows you to throw more samples on your lane or sequence deeper if you need to call some low variant allele frequencies. So this has been around for a while. So Twist came into a semi-crowded space. And so we had to really differentiate ourselves. And so one of the key things that we looked at was improving the efficiency. That is the best way to drive down your sequencing costs, is by having your target enrichment perform very, very well. And so the first thing we did was we shipped double-stranded DNA probes. We'll kind of go over what this looks like visually, but the idea is that you're able to capture the plus and minus strand and increase your library complexity and your capture efficiency. We'll go over what uniformity means in a second, but it's the idea that your sample is not over or under sequenced. So we really focus on that metric, which a lot of other companies tend not to do because it usually looks pretty bad. Finally, we also NGS QC all of our panels. So whether you're doing an off-the-shelf exome or any of our methylation panels or a custom design, they'll all be NGS QC'd. So you are getting actually what you've ordered. Our entire workflow is very flexible. So from the individual components of the kits, whether library prep or target enrichment, to the size of the panels, if you want to do a custom design, we can do a couple hundred probes up to thousands and thousands of probes to really fit your needs. And at the end of the day, this is all to lower sequencing costs. So putting more samples on your flow cell or sequencing deeper for those difficult to call mutations. So when we talk about uniformity, this is kind of the example of a non-uniform target enrichment, where you have these three targets here, these gray blocks, and you have your call pileups on the graph above. And there's a line for like 30x coverage, a pretty standard kind of exome depth. So anything in that hashed green area is over sequenced. So this might not be necessary for calling any of your mutations in those targeted areas, and could have been more efficiently spent on that third target here that is significantly under sequenced. So these reads here are again wasted because they are not reaching that 30x depth and therefore are unusable. And then you always want to avoid your probes binding to an off-target region and capturing something that you don't actually want to sequence. So we have managed to kind of alleviate a lot of this over and under sequencing through a number of means. And so the first one would be the fact that we actually GC bin all of our probes. So we take a look at the GC content in the individual probes, and we bin them based on certain GC percentages. And this will bias the amount of probes in your panel. And then with the increased efficiency of high GC probes to hybridize, it'll balance out to an even capture. So you go from this massive amount of over and under sequencing without any of this balance GC binning to a much more even capture, where you're getting the same amount of data piled up over each of your targets. I mentioned this earlier, but we also ship double-stranded DNA probes. This is just kind of a visual representation of that. Other companies will send single-stranded DNA or single-stranded RNA, which is leaving half of those DNA molecules in solution. And you're not pulling those out, which is kind of wasted library complexity and data that could be used in your sequencing run. We have NGS Space Quality control. So this is the idea that we're going to throw this on a sequencer of your panels, and you're going to get exactly what is supposed to be in your panel. And you don't have to worry about missing any of your data, even lot to lot. And the best way to kind of compare how this target enrichment system compares to other companies is to look at exomes. Since everyone has an exome, it's the most apples to apples comparison that's possible between systems. So we actually contracted 2 third-party labs to take a look at our newest exome, which is the Exome 2.0, compared to 3 other competitors. This was performed to vendor spec. So we followed all of the protocols, or these 2 third-party labs followed all these protocols, 50 nanograms of input for all of them except for vendor K, which was 100 nanograms of input. All samples, all data was downsampled to 6 gigabases to level the playing field. And the sample was NA12878. Looking at the kind of most important capture metrics to evaluate how well a capture performed, the first is a measure of uniformity. It's the fold 80 base penalty. It is essentially a measure of how over and under sequenced your sample is, where 1 would be a perfectly uniform capture, where every single target received the same depth of coverage. And then as you go from there, it's a more ununiform capture. So Twist is sitting at a 1.28 for our Exome 2.0, which is exceptionally good for target enrichment if you've ever performed one of these experiments where all of the competitors can't really come close to that sort of uniformity. And this is across our exome, and you see this in custom panels as well. Very good uniformity. The other three metrics are more self-explanatory. So % on target. This is how many of your probes landed on the target of interest and not elsewhere. So the opposite would be off-target effects. %0 coverage targets. So these are the targets that have no calls piled up over them whatsoever. And then the final one is looking at, again, 30x coverage, a pretty typical exome depth, and how many of your targets reach that threshold of 30x, with Twist again leading at 97.68%. The GC binning is really evident also in these experiments. When you look at the normalized target coverage across the GC spectrum, the GC content of the targets. And you see Twist is very tight within this small window and flat across that spectrum, whereas you can see very large biases towards that high GC because of the efficiency of pulling down those targets. If an exome, again, is not not of interest of you, we have a very robust customization of panels option. You work very closely with field application scientists such as myself, as well as a bioinformatician, to design a very personalized custom panel that will only pull down your targets of interest in whatever genome you want, and targeting pretty much anything in that genome. Once you provide us with whatever targets you have, so this can be gene names, genomic coordinates, SNP location, CPG sites, kind of any information that you can provide to us for your targets, we can design a panel around that. We will build the panel for you. You can order it, run a kind of pilot experiment, if you will, and we can actually ingest those FASTQ files from the sequencer, and we can take a look at how the capture actually performed. So look at these capture metrics that I presented a little earlier and see if there's any tweaking we need to do on the bench to maybe change some temperatures to really increase on target or drop AT dropout and things like that to make your capture as efficient as possible. We do offer kind of a pretty wide suite of products, everything from kind of post-nucleic acid extraction till you load it onto your sequencer of choice. So we have multiple library prep options. We have enzymatic fragmentation, mechanical fragmentation. We do offer UDIs as well as inline UMIs. And then on the target enrichment side, we have all of the enrichment reagents, all of our catalog of panels, our custom panels. And these can be used to target DNA, RNA, long reads, methylation. So we're kind of agnostic to what you're trying to target and have a solution for almost anything. I'm going to move into specifically talking about our methylation detection system, which is probably a good point to stop and see if anybody has any questions kind of about the general background of our DNA target enrichment system. Okay, I don't see anything in the chat, so I'm going to move forward and I'll take questions again at the end of the talk. So Twist really moved into the methylation space, trying to provide a better solution for next-generation sequencing-based epigenetic studies. And the idea was to provide all of this exceptional uniform capture and sensitivity and flexibility that we have in the DNA space, but apply it to epigenetics. And how we did that is we partnered with NEB to provide their NEB Next EM-seq kit that's been optimized for Twist target enrichment. So the conversion and library prep will go through this EM-seq kit, and then target enrichment will fold into the Twist fast hybridization system with some optimizations for specifically post-conversion DNA. If you're unfamiliar with the NEB EM-seq kit, it is a much gentler way to convert your non-methylated cytosines compared to something like bisulfite. So it's very gentle on your DNA, which allows you to put a lot less material into the assay in the first place, as well as allowing you to detect more CPG sites. It's more uniform across the GC spectrum, and it's a very efficient workflow. So you'll be able to use this on cfDNA, FFP, kind of those difficult to run samples. The conversion rate of this kit is very high. So bisulfite being the gold standard, we usually compare against that. So conversion rate's about the same. So 99.7, 99.8% conversion. So you'll be sure that you're actually going to call your methylation statuses correctly. But again, the benefit is it's not going to destroy your original library molecules. So unlike bisulfite, you just out of the box get much higher library yields. So again, you start with a lot less, allows you to do these cfDNA applications. And speaking of cfDNA, one of the benefits of enzymatic conversion is it maintains the fragmentation patterns of cfDNA. So if you're doing these studies in blood, you're looking at the cfDNA, you'll maintain those nucleosome turns and the fragmentation pattern and not destroy them like you would with bisulfite treatment. And this allows you, since you're not destroying a lot of your DNA, it allows you to actually call more CpG sites. So you're looking at 15% more CpG sites on average called with the enzymatic conversion, but the core of the calls are very concordant with the bisulfite treated samples. And not only does this improve your upfront library prep and conversion steps, but it actually translates to downstream improvements to target enrichment. So again, looking at these kind of target enrichment specific metrics, you get increased 30x coverage, increased uniformity. You're going to have lower off target and lower duplication rates. So you're getting a better sort of conversion and library prep stage, as well as a better capture downstream. So that's kind of why Twist decided to go with this enzymatic conversion compared to something like a bisulfite. I mentioned that there isn't this kind of fall off that you see in high GC regions that you see with bisulfite. So whether you're looking at a hypomethylated or hypermethylated gDNA, you don't have this very steep drop off once you get to high GC content. It prolongs that coverage kind of out further into the higher GC targets. This is all to make everything a lot more efficient and actually be able to call in these areas, as we'll see a little bit later when we look at some data that's been run through these kits. One of the decisions we had to make early on was whether we wanted to do pre or post capture conversion. And this was a very important decision to make, as a pre-capture conversion allows you to lock in all those methylation states and all of their different levels. So your DMRs, whether it's a 50% methylated, a 75% methylated site, lock that in and then do capture after that, allowing for an intermediate amplification step to bring that library up. So further reducing the amount of input you need to the workflow. The post-capture conversion yields a lot less complex libraries because you're doing the capture as like a sort of DNA-based capture and then post-capture converting. So you lose a lot of that complexity that might be in your original sample. Now, how we tackle the increased complexity of the actual probe design for pre-capture conversion is we look at each individual CpG site and we consider it to be fully unmethylated or fully methylated. And then this is all in an in silico algorithm. It goes through conversion and then PCR. And in this final post-PCR state, that's where we do all of our probe design for each CpG site. So you're getting 8 pieces of DNA to target each individual CpG site. And then with the efficiency of our probes to capture mismatches, you get the ability to call things that are unmethylated to slightly methylated with the unmethylated probes and the opposite with the methylated probes. So it allows you to call DMRs a lot more efficiently. And this algorithm we're always trying to improve it. So this is just an example of improvements we've made to our machine learning process, our algorithm to actually design the probes. So again, you see with the default kind of design and it's how efficient it is. And then with optimizations that we've made to our algorithm for specifically methylation designs, we've improved all of the standard metrics, whether it's uniformity, off target, duplication rate, everything like that. To really show the power of this increased library complexity, we developed a contrived experiment where we took 0% methylated cell lines and 100% methylated cell lines, and we mixed them at different ratios to have a contrived sample of 25%, 50%, 75%, and 100% methylated samples. On the left here is just, again, those same those target enrichment metrics and how each of those samples, each of those target enrichments performed. And on the right, you have methylation calls in IGV. And the nice thing to see is that the 100% methylation, 100% methylated samples are being called at about 100% methylation. 75% looks like 75%. So they're all decreasing appropriately. And that's something that this, this pre-capture conversion allows you to achieve. The reproducibility is also very high with NGS, which is something we'll get into in a second about, methylation arrays and their kind of weaknesses. This is 8 replicates of Na12878 in a 8-plex capture, and it's very high concordance from sample to sample with, R values of 0.98 in general. So very high concordance. When we released this kit, we, had a good opportunity to address some difficult to, difficult samples and experiments to run because of the current technology. So we actually released our Human Methylome panel recently. And this is a panel that's kind of going after the array technology, something like the 850K. It's a very large panel. It's 123 megabases large. It covers about 4 million CpG sites, and that's direct coverage with shadow coverage due to the reads of NGS being, different overhangs. You reach about 6.5 million CpG sites. On the right is just a breakout of where these CpG sites lie, whether they're in CpG island or a shore, shelf, or sea. And if you're wondering what it kind of overlaps with the 850K, since this is trying to directly target customers that are currently using that array, we cover about 3.1 million more CpG sites directly. Again, you get another about 3 million with the shadow coverage. So we cover about 95% of the EPIC array. And these CpG sites that were left out, these 44,000, were generally CpG sites that weren't robustly called in the array anyways, or they were deemed as non-medically relevant, so they were removed from the panel. But some of the key advantages of running a methylation experiment over a microarray is increased dynamic range, which we'll see in a second, as well as the ability to, say, call SNPs if they're at a high enough allele frequency. The reproducibility is much better, the ability to run FFPE samples or fresh frozen samples, and as well as the flexibility that we have to rapidly change the content you're looking at. So whether you want to do this large methylome or a much smaller panel that's just targeting your favorite CpG sites in your lab. Since this is such a large panel, it's kind of hard to think about how much sequencing you have to throw at it. So we have done some empirical studies to kind of give everyone an idea of what they can expect sequencing this. So this is on the x-axis is the raw coverage, and then on the y-axis would be the post-alignment coverage or the mean target coverage. So we usually advise that 150x raw coverage is the sweet spot, because post-alignment and then looking at mean target coverage, it ends up being around 50 to 60x coverage, which is good for most experiments. But this gives you an idea of if I throw X amount of coverage at my sample, I can expect Y amount of post-alignment coverage. The good thing is, though, no matter what of these depths you're actually going to, you're not going to see major changes in the per site CpG percentage. So this is just, again, 100, 150x, 200, and 250x overlaid in the BRCA1 enhancer region. And you can see the methylation calls don't vary that much, no matter how much sequencing you actually throw at the samples. Similarly, you're going to align a lot of your reads. So 93% of reads align. So this was using PWA meth on a NovaSeq with 2x151 kits. So again, you're going to get more reads, obviously, the higher depth, but you're going to get the same, pretty much the same percent of them are going to align no matter your depth of coverage. To really illustrate kind of how the data you would get from this EMC kit with our Human Methylome panel, we compared it to the EPIC array. directly. So this was called with Illumina Epic Array for the arrays, and then the Twist Methylome was run through BWA Meth, Methyldactyl, and then with the parameters seen here. And we only compared the overlapping 816,000 CPG sites so that we could see how they directly performed. So there's a very high correlation between the methylome and the Epic Array. So we have an R value of 0.973. So very, very accurate compared to the array. One of the things we noticed when we were looking at this data that we found once we downsampled here on the right is there's a pileup of these CPG calls in the methylome at 0% and at 100%. And when we did a density plot of all of the CPG calls in the experiment, what we found is that the Epic Array not only has a bias to lower percent methylation calls, but it also has a much narrower limit of detection. So the Epic Array was not able to call 0% or 100% methylated regions very well, which really biases the data inwards as well as to the left or to lower percent methylation calls. So in general, you're able to call these truly 0% or 100%, but we also found some interesting sort of single cases that could be more prevalent in data the closer you look at your calls. But this is a comparison of the methylome and Epic Array data at the BRCA1 promoter region. The methylome there is in the middle in green, and the Epic Array is in orange. The one thing you will notice is that the methylome is calling on general a much lower percent methylation at each site, so 1%, 2%, whereas the Epic is more 2, 3, 4%, so much higher methylation calls. And then we saw interesting cases like you see in that red circle, where the methylome is calling 0% methylation and the Epic Array is calling 18.5%. And since it's NGS, we can drill down into each individual read that was called over that CPG site and actually see what is going on there. Is the methylome just not able to sequence that area? What is the difference? And this is actually a known C-to-T mutation that is found at this locus in the BRCA1 promoter region. And so this shows the power of NGS versus an array-based approach, and that you're able to maintain this SNP detection because you're calling both the plus and minus strand in each of the reads. And so you're able to detect these as long as the variant fraction is above about 30%, you're able to call these, and you won't call them as some sort of non-methylation event with the mutation. So I hope everyone learned a little bit about our methylation detection system, how Twist is kind of pushing the envelope when it comes to target enrichment into all of these different new technologies, whether that's new sequencers, the long read sequencing, RNA, methylation. We're always looking for customers to help us push this technology as well. So if you have an interesting project, please reach out to us. We would love to hear about it and see how Twist as a company can help you achieve that. We have all of these kind of solutions to date for all these different type of targeting sequencing, whether you're looking at cfDNA or anything to ag bio in infectious disease. With that, I would love to take any questions that anyone might have.
Well, great. Thank you very much. I think Lynn has a question. If you can directly read it and address it.
Yeah. So Ling is asking, can you address sample starting amount and quality, particularly for cfDNA and FFPE DNA in more detail? So minimum requirements. Yeah, so the EM-seq kit from NEB, it says that about 200 nanograms goes into the library prep kit. If you read the EM-seq kit specifically for cfDNA applications, they say it goes down to about 10 nanograms of input. So that would be kind of the range you're looking for. With any sort of sequencing experiment, the more DNA you put in, the more unique molecules you have. So if you go down to 10 nanograms, you have to keep in mind that you might see more duplicates or things like that. As far as quality, again, the higher quality, the better. We've had customers with, if you're familiar with DIN values, down in the 3 DINs of 3 can still get some pretty robust data. But in those sorts of circumstances where you have that more degraded DNA like FFPE, you're going to want to try to put as much into the system as possible. So try to hit that 200 nanograms of input to ensure that you're getting a robust library out of the EMC kit.
Great. I have a question regarding the assay itself. So, and do you have extensive PCR steps in the library preparation protocol? And so some of the quantification of that, will that be driven by the DNA replication that the same molecule are being sequenced multiple times?
Yeah, exactly. So that's part of the reason when you put in 10 nanograms of input, you're going to have to push those PCR cycles up. So at the end of the day, you're going to be sequencing more duplicates. So if you can have more DNA going into the system, you can limit this to some degree by trying to keep the PCR cycles as low as possible, but you need to have enough material out of library prep to go into hybrid capture. So as long as you can hit that, you can kind of limit that duplication rate.
Yeah, I guess the effective coverage that you are putting out there with 150 million reads, that is that 40 or 50x coverage? I don't remember exactly in one of your slides. What are if you think about removing the DNA duplications, and what are the real coverage for that?
So this data would be post deduplication. So this is taking into account that now this specific experiment would have been run with the input of being 200 nanograms, and it's run with NA12878. So it's really high quality gDNA. So this is kind of best case scenario. So everything else would need to be kind of subtracted from this to get to what like a FFP or a kind of in the lab sample would look like.
Yeah, but the effective coverage, so you have already removed the duplication. I mean, the rays that coming from the start and end at exactly same locations of multiple rays, they will be removed at this point, right?
Yeah.
Okay.
Yeah.
All right. Charlie, go ahead.
Yeah, thank you for the great presentation. I have a question regarding the different species. Can you use the same technology for mouse or rats DNA samples?
I believe so. I haven't personally used it on any mouse or rat, but I'm fairly confident that you can.
Do you have the capture for the mouse and rat genome?
It would need to be a custom design, but we design in a mouse and for the DNA sequencing. So if you give us your reference genome, whether that's MM10 for mouse, we can design in that space.
Yeah, let me follow up a little bit on the technology. So can you pull the samples for sequencing? I mean, do you have like a unique barcoding for each molecule? Or I mean, what's the strategy for pull the samples?
Yeah, so currently with the NEB EM-seq kit, we have 96 barcodes, so you can pull 96 samples. Each one of the captures that you would perform goes through an 8-plex. So you would take 8 samples, pull them into one tube, do a capture on all 8 samples, and then once you're finished with all of the target enrichment and PCR steps, you would pull a number of those 8-plex captures together onto a flow cell. So you can multiplex up to 96 into a single lane.
So the deconvolution would be pretty much the same as other pooling algorithm?
Yes, correct. Yeah, the demultiplexing is the same.
Oh, okay.
And for this one, I say that you don't have a UMI option, right?
No, currently we don't have UMIs because when in the actual protocol, when you go through the conversion, it's post adapter ligation, and our UMIs would be like in line, and we just don't have methylated UMIs currently. The NEB adapters are methylated, so they don't change, so they don't mess up the priming reaction.
Yeah.
Yeah.
Okay. Do you have RNA capture protocol?
So we the protocol doesn't change as long as you can get the RNA into cDNA. So as long as you have that RT reaction, you can get it into cDNA, it would go through the same kind of library prep and target enrichment. And we can do the custom design in RNA space. So if you need to do like novel isoform detection and things like that, we can design towards that sort of RNA output.
But the RNA has a lot of very high dynamic range, and yeah, I don't really know how that will work. So you don't have, do you have a protocol that you support from like the RNA, the total RNA for the low throughput, low input and the low quality RNA and go to the target?
So we have some protocols. They're specifically written for more like viral detection for RNA viruses. I will say that we will be soon have some RNA solutions that would have a more dedicated protocol for that sort of application. But the current design process would be pretty much identical to what we have in the future when we have an RNA solution.
But is that going to be single-stranded or double-stranded capture?
Sorry, so it won't be direct RNA capture. It'll always have the cDNA synthesis step and then go through that. It'll be, but it'll be the same double strand, getting the plus and minus of the.
I see.
But it's going to be cDNA.
All right. Okay.
Can I ask?
Yeah, go ahead.
Oh, okay. Thank you. So I'm just curious because as a investigator, I'm curious whether your the kit or the reagents are primarily customized for each investigator, or this can be generally used by the core facility. I'm just curious what's the interface here. Should the investigator do all the reactions, then deliver the library to the core, or the library can be made by the core facility as a service?
I think that might be a better question for you, Long, but
what's the design here?
Yeah, so we do, we can handle both. So some cores like us to send the panel directly to them and they will do all the target enrichment, but we also can ship directly to your lab to where you can do the library prep. It's really up to the core facility to decide what that is. As far as the panel content, all of our off-the-shelf panels can be ordered by anyone. If you have a custom design made for you or another investigator, we can print that for the other person as long as we get permission from the investigator. Your panel that you design will be your proprietary design, and we won't share that with anyone else unless you let us. But yes, the core facility could handle the library prep and target enrichment.
Yeah, just curious the kit size. For a starter, investigator, they might want to try 4 or 5 samples at a time. What's the kit size? You can do let's say 36 or 96 reactions from each kit, or what's the kit size?
Yeah. So if you're ordering off the shelf panels, so say like a Methylome or something like that, we have smaller kits where you can get 16 samples worth of capture. If you're going to do a custom design, the smallest order for a custom design panel would cover 96 samples. Yeah, so that, and then the pricing is depending on how many samples you're going to do and that sort of thing. It drives down the price if you do a larger project, that sort of thing.
Yeah, I think those are going to go with the individual discussion. If you are working on the human exome, and the chances we probably can order larger kit, even you don't use all of them. But if you do the exome capture for rat, for example, and we don't get a lot of those requests, probably we need to order smaller batches, or you have to order, be responsible for all the reagent that you order. If it's customized, that only you're interested in, you have to have 96 samples. Otherwise, it will be more expensive per sample, I guess. Oh.
Thank you.
All right. Any other questions?
I did have a second question regarding the comparison data. Tim, do you have any on the comparison data of different quality of DNA?
I would need to check. So in general, we typically stick with like high quality genome in a bottle for a lot of our R&D work, because it's kind of obviously the best. It's gonna be the best data and the most reproducible. I would need to check to see if we have customers that are willing to share their DNA quality input and output. But that's something I can look into to see if we have if another FAS has customers that are already sharing that kind of data. I can get that to you.
That would be great. Thanks.
Thank you, Tim, again for your presentation, and thank you everyone for coming.